Hey gang, so let's do a little bit of review for the lab final. We got a study guide we can run through quick and a couple slides. Now, the slides I posted on Brightspace, they are essentially a lot of the same slides that almost all the same slides, I should say, of what I posted all throughout the semester. So don't bother printing them out, just kind of go through the stuff we've already printed out and kind of go through them. But these are the slides that will most likely get used on the lab final. I know it's a lot, but hey, it's a big test, so there's lots of options for me to choose from. But let's do the slides first, actually. I'm not going to go through every single one. I'm just going to go through some of the tissues and things like that that I think we do best by reviewing those. So let me bring this up, and we shall show that. So bring this screen. All right, so here we are. And like, as I said, most of these slides are things we've seen before. They're exactly the same slides on all of our other review slides. So make sure you go through them. I said the most things I want to kind of worry about are the histology slides that we might be a little bit more unfamiliar with. But everything else, as I said, these are literally copy and pasted from all of your other practice slide packets. So make sure you review these in particular, and you should be all set. Once again, as to the tissues, all right, so respiratory tissues, make sure you are able to recognize a trachea. And here's the thing, gang, what's really going to be is you're going to see this and it's going to ask you, okay, what part of the respiratory system this is. You see pseudostratified ciliated columnar, you see some hyaline cartilage down there at the bottom, down here. It's trachea. And here's kind of another shot where you can see the pseudostratified ciliated columnar up at the top, and then the hyaline cartilage down at the bottom. If you see that, it's trachea. That's what it comes down to. If you go back through our lab PowerPoints, you might see some slides of like lung tissue with the simple squamous epithelial tissue. See that? It's lung tissue. Like nothing crazy, but review those things just so you're not surprised if they show up on the final. That sort of thing. All this stuff, as I said, you've seen before. A couple other things, digestive tissues. All right, so like I said, in lab, when we actually were doing digestion, this is the liver. This is the perfect liver, okay? You see something with a hole in the middle and it looks like a hexagon. That's liver tissue. Here's the thing, though. The chance of this actually showing up on a tissue slide, and we use actual tissue slides during lab, is like slim to none. It's more going to look like something like this. So you can still see the central vein in the center. You can still see the little sinusoid capillaries going off to the side, but you don't get the perfect hexagon. It's going to look more like this. So if something that looks like this with a hole in the center like that comes up and they ask you what sort of digestive tissue it is, it's going to be the liver. Okay. Other than that, liver's a good possibility. You might see intestinal. So you see the villi. You see something that looks like fingers with some simple columnar in there. You might be able to see a brush border of microvilli in it. You know that's going to be the intestine. So just like this. Same sort of thing. See something that looks vaguely like this? Small intestine. That's the one that might possibly show up is pancreas. And usually the slides are a little bit darker, more of a bluish color, but or purple, but you'll still see kind of darker, denser tissue surrounding like a little island, thus pancreatic islet, of lighter color tissue in the center. So once again, that's going to be pancreas if you see something that looks like that. Other than that, gang, like it's literally everything we've done before, and I don't really see any point in just rehashing all that when it'll be time better spent on your own just identifying everything, and you already have the slides set and filled out and hopefully ready to go from our previous reviews for each quiz. I do want to go over, kind of take the time to go over the actual printed study guide with you, just once again, most of it is going to be, okay, what's this on this picture? What's this on this slide? What's this on this model? Things like that. So nothing we haven't literally done before. But let's take a look at the printed study guide just so we can go over some of the more important stuff. So let's bring that up. Take a look. There we go. All right, so here we see the printed study guide. Remember, your final lab practical is 50 questions. It is multiple choice. It is Scantron. The hardest part of this test is going to be literally making sure you're filling in the right bubble on the right number, because it is a Scantron. So special senses, once again, the diagrams of the external and internal eye structures, the structure of the retina. So once again, make sure you know the difference between the rods and the cones and the ganglion cells and the bipolar cells and things like that. The eye dissection, once again, look through the slides. Olfactory anatomy, so that's the olfactory epithelium and like the actual olfactory epithelium with the olfactory bulb and the sensory cells and all that good stuff. Make sure you're able to identify all the parts on that. Same thing with the inner ear and the spiral organ. Ear models are going to be on here. The spiral organ model is going to be on there. Things like that. So make sure you're able to identify all the anatomy on those. Things like that. What gives students the most trouble on the lab final, I would say, would be the endocrine system. It's 3 questions. It has to do with our hypothalamus, pituitary, thyroid axis. And remember how we did the little rat experiment with like the shivering and things like that. And sometimes we gave the rat propylthiouracil, which blocks the use of iodine, so the poor rat can't make thyroid hormone, which reduces its metabolism. But also, remember we talked a little bit about how a rat would react depending on what we did to it surgically. So we talked about this in class. Remember, actually, let me switch to a little whiteboard quick. This might be a little bit easier for us to kind of do if we do that. Let me bring up a little Wacom board here. Rusty. Oh, there it is. Okay, so let's bring up our little whiteboard really fast as a review. There we go. We'll close the share. Bring up the whiteboard. Share that. Wait. There we go. So remember back when we were doing this, we had our hypothalamus, right? Which makes thyrotropin releasing hormone, our TRH. That goes to our anterior pituitary. Which in turn releases thyroid stimulating hormone, or TSH. And that's going to be go to our thyroid gland. And that's going to release thyroid hormone, TH, right? All right, so remember if you do propylthiouracil or PTU, we're still going to make TRH, we're going to still make TSH, but we're not going to make thyroid hormone because PTU blocks iodine. And remember, iodine is 100% necessary for thyroid hormone production. If we surgically remove the thyroid gland, once again, we'll still make TRH and TSH, but we can't make thyroid hormone. If we surgically remove the anterior pituitary, we can't make TSH, which can't stimulate the thyroid gland. So that means we can't make TH, thyroid hormone, things like that. So once again, just kind of review that stuff. Remember think about what would happen to which gland and their secretions depending on different factors, things like that. So that's kind of like one of the harder things to kind of remember and go over. I said we only did it one day, and it was way back at the beginning of the semester. So it's one of those things you should just kind of be aware of and be prepared for on the lab quiz next week. Blood, identifying leukocytes, blood typing. Okay, so once again, remember what the different blood types and what the different agglutination, the clumping reactions mean. So for example, anti-A antibody only agglutinates something with A antigen. If it's got A antigen, it's some sort of type A, either type A or type AB. It's not B, it's not O. So if it agglutinates with anti-A, it's either A or AB. If it agglutinates with anti-B, it's some sort of B. If it's only agglutinating with anti-B, it's straight up type B, okay? If it agglutinates with anti-A and anti-B, it's AB. If it only agglutinates with anti-A, it's type A. If it doesn't agglutinate with either anti-A or anti-B, it's type O, okay? So right, type O doesn't agglutinate with anti-A or anti-B. And if it agglutinates with anti-RH, it's Rh positive. If it doesn't have agglutination with anti-RH, it is Rh negative. So remember that. Think about who can be transferred to who, what can be transferred to what, that sort of thing. Heart, once again, look over your diagrams. It's the big thing. Same thing with blood vessels and blood vessel identification. So remember, go through those slides. Anatomy models, the histology slides, definitely be able to recognize the trachea. Respiratory capacities and volumes. Once again, that diagram we had on our last lab quiz on respiration. So with the vital capacity versus inspiratory reserve versus expiratory reserve versus tidal volume versus residual volume, what adds up to vital capacity, what adds up to total lung capacity, that sort of thing. Digestive system, remember our tunics of our digestive system walls, starting with our lumen, then we have our mucosa and submucosa, the muscularis, and serosa. Make sure you're able to identify all those. Definitely check out those histology slides again. Be able to recognize if something is the liver, the pancreas, or the small intestine. Make sure you know all your fetal pig dissection stuff. Urinary system model system. Remember your models, the anatomy diagrams, nephrons. There will be acid-base problems on this, so make sure you know that. Chemical controls of respiratory rate. So think about how your respiratory rate would be affected if pH increases or pH decreases, or carbon dioxide levels increase or carbon dioxide levels decrease. So obviously if CO2 levels increase, we're going to increase our respiratory rate and respiratory depth to get rid of that extra carbon dioxide. If CO2 levels decrease too much, we are going to reduce our respiratory rates and respiratory depth to get CO2 levels back to a normal homeostatic thing. Remember, that goes hand in hand with pH. Remember, if pH gets too low, increased CO2 decreases pH. So we're going to try to breathe harder, breathe faster to get rid of that excess pH. If pH gets too high, right, too little CO2 leads to a high pH. So what we're going to do is we're going to reduce our respiratory rate and our respiratory depth to increase our CO2 levels to drop our pH down to a lower pH level. Okay, so make sure you know that. Look over, and this isn't really on the lab quiz, but it's more of a lecture exam thing. Look over the buffer. So remember, we have our bicarbonate buffer system that works in the blood. We have our phosphate buffer system that works in like the extracellular fluid and inside the cells and things like that. So just kind of review that little PowerPoint really quick. Male reproductive anatomy, female reproductive anatomy, stages of development, things like that. Once again, the diagrams. Make sure you know that stuff and you should be fine. So as I said, were we doing this in class? I do this all for you, but since we're not, and I still want to record a really big lecture final review for y'all, I'm going to save my time and do that, since I might be going into the hospital this evening. So good luck, Godspeed, may the force be with you, and hopefully I'll be doing this in person on Tuesday with you, but if not, just a couple of quick hints and tricks for doing this. So thanks, guys, and I will hopefully see you soon.